Okay, here's the plan. We're going to have each of the rapporteurs from our different groups come up and give a very, very brief summary of what happened. 5 minutes, we're going to wave our arms and jump up and down and say, okay, stop at 5 minutes, and then we're going to continue our discussions outside with the wine and the food and the whatever, one-on-one, because we have been sitting in this room for too long. So, okay, group one, is that blue? I forget what sequence we were down the hall. We're going to start with red. Okay, here comes red. 5 minutes.
Okay, so I wanted to start with the first question. Is this on? Okay. So the first question was really about the different built environments and whether or not we can really look at those together and compare them across the different built environments. And there was two senses in the meeting. One was that it's premature, that we don't know. There's too many different variables, that we should be looking at them in as different spaces, that they are being used by different cultures, by different people in different ways. The architecture, the functionality, and how the buildings are used is something that really drives the differences. Also, the indoor-outdoor effects. Because they're in different geographical locations, the effects differ, and they also differ by building type. So whether or not things are, whether there's ventilation systems, and whether it's coming into the building or not. Also by things like pets and other humans entering the building. The other sense was that we can move towards a better understanding. So we're talking about using mechanistic versus just survey studies. So actually making a change to the environment such that we can see how it is affecting it. That sense led to this idea of if you control all the variables, such as occupancy or if there's pets or sex or any of the possible variables, that the building type might not matter. So the concept of everything is everywhere, but the environment selects was brought up. In addition, moving on to the second piece, we talked a lot about the sampling and analysis and methodology. There was a sense that there was too many parameters. The collection of metadata is very difficult, and that it's done differently by different people. There's also a question of temporal variation. A lot of the studies take one instance. A lot of the studies take several instances at several months or even years apart. There was a feeling that there's no consensus through sampling methods, bioinformatics. A lot of the discussion was talking about epidemiology, whether there's disease or exposure, and how we are looking at the samples. So essentially we're talking about if there's a disease state, then we look at that disease state or and the associated microbiome. Or we look at the exposure and see where that disease state is absent. So a lot of the sample methodology was brought from an epidemiological state or point of view. One of the things I noticed is that there is a big difference between the profession of the people who are there. So if you are an engineer or a building scientist, you are thinking in terms of that direction. If you are an epidemiologist, you're thinking in terms of that direction. If you're a bioinformatician, you're thinking of in terms of that direction. And so there's a lot of differences between those, and it seems like it's hard to bring a consensus based upon that. A big point that was brought up is that reference methods and sample methodology should become an agreement. People who are working in the health fields talked a lot about there's reference sampling, that the sample is taken specifically 1 foot from the corner of the wall, or those type of things. And it seems like that's something that we need to drive towards for this. I think that's about it for me. No question.
Okay, great. So just a couple things covered from the blue group. The first question on the results of the building type, if it can be applicable. Really, we thought this was kind of based on a building science approach here on looking at these. And we could said it's certainly you can compare building types and probably easier in something like a hospital than to compare across different building types. And definitely as you have more data sets, then you would have larger power to run your statistics, but generally that'd be hard. We came up with kind of 3 generalizations across the different building types that increased ventilation is good, increased indoor dampness is a bad thing, and urban air is probably not so great for many different situations. So those were, I mean, out of all the things we talked about, those are the only kind of 3 generalizations we had from the building types. We also looked at or discussed if this is comparable. So if we did get to the conclusion that this would be comparable across building type, then we should be able to test this with a model and should be able to fit it with parameters. And then at that point, most people didn't feel very comfortable to be able to do that or have seen that before in literature. Probably more likely that we could test these types of models, or we might be able to get there with specific species or maybe genera, but not the communities in a whole. And then if we do have, though, information on ecology, we did say there is some bright news there where you can probably do some prediction and some modeling about future results if you have the information already. So what might survive there, what might last there, what might thrive in given these different situations. So that was question 1. Question 2, we noted that several speakers had said they had some, there's certainly some issues there with comparing results across different sampling and methods. Same as the first speaker here. And there could be errors. We noted that the errors could multiply. And so as you have errors, you can actually go further and further away from maybe it would be the standard. If you have, it was noted that if you have enough of a difference, then you can actually, you might be able to still see the microbial communities might be different enough that sampling bias or DNA extraction bias or whatever other bias you have, you still can see kind of the main things that are main drivers that are happening to the communities. But we weren't completely clear on that one. Best practices, we thought NIST is a great place to start, and likely their work is going to be important in our field. If they can standardize some sampling, extraction, sequencing, and analysis. We did note that, at least from our group, a lot of us felt comfortable probably being able to compare our own studies and if we, and our own extraction methods, and then definitely the comfort level went down after that when you start thinking about comparing to other people's samples, not in your lab or not standing next to you, or not you. But it would be great if we had that. You do actually, though, we got to think about, it might be important that you, as you get to standardization and you're driven to standardization, it was brought up from the Gilbert study on the microbial, what is big study on Earth microbiome, I guess they call it, that people are driven to that maybe for bad reasons. So you'd say, well, I'm going to do the Earth microbiome DNA extraction. It might not be the right standardization for your question, but people do it just so they can have you can do the standard method. So there is some danger with the standardization. And then lastly, we talked about the different types of buildings, and we definitely thought that material could have an effect on the built environment. For example, the leakiness of a building, the humidity, the different walls types. It has building use as something that could have a big effect. One thing being we cited was occupancy in areas. Humans, the interaction with surfaces has a change, or is changing probably with how much we interact with surfaces or don't in the built environment. And as more and more people kind of learn about this, the built environment, you see more people flushing the toilets with their feet and stuff as they learn and more about microbiome stuff. We also said there's some non-standardization for like ventilation strategies, which you call natural ventilation for one engineer might mean a very different thing for another engineer. Again, getting to maybe this terminology stuff. And then we also highlight in this one with the different types of buildings, it's important to have the metadata. If you don't have that, then it's what you see could just be noise. Third group? Or there's only two?
Thank you. Yeah, it's the Crystal Group led by Jessica Green. Yeah, so we initially split this discussion up into two aspects. One is the technology gaps versus the knowledge gaps. And the first question that was addressed was the third one, whether there were inherent differences between types of built environments. And I mean, obviously the consensus answer was yes. And in terms of the technology gaps in that regard, one of the key points raised was we need to differentiate between the dead versus live fraction of cells that we're examining in the built environment, especially in the context of the huge amounts of genomic data that we're generating. Again, in terms of defining the syntax of names that we provide to the data sets, there has to be consensus because not all the built environments have air conditioning and the ventilation conditions that is ubiquitous to what we have specifically in the United States. And in different continents, there might be different conditions. And we talked about the open air hospital settings where, again, it's not a closed environment and there's going to be an obvious variation there. So looking at that, it's also sensible to think of the building materials that would exist in such open environments and whether it's feasible to sample in such differing environments and how you can standardize sampling methodologies for different surfaces and the challenges that come with those methods. Like, for instance, using a swab-based collection technique and retrieving the amounts or the yields of DNA required for sequencing. That is, again, a huge variability that can transpose between different continents or different sampling conditions. So the other aspect that was talked about was, again, the challenge of sampling and also to, we need to look at the new methods that have been developed. The one that was mentioned was by Jeffrey Segal, where they are using an enhanced swabbing capacity that they have brought in terms of the sampler that they've developed. And the other aspect was to look at the approach of having the idea that each of these habitats could act as a habitat patch or an island where each of these patches have their own unique environmental hotspots. So those hotspots need to be examined or specifically looked at the cohorts that coexist in such hotspots. So the other aspect that was discussed was looking at adding a specific set of variables that would be measured that are similar to epidemiological studies, and such variables would be like age, sex, race, and also habits like smoking, etc. I mean, something to find like an analog of that. I could think of an example in an ICU setting where you want to examine if an infant has been treated with antibiotic, or even the mother in that case, and looking at the effects, after effects of whether they're sharing in terms of microbes of such infants. It's just an example there. Again, and the ecological principles of addressing the knowledge gap, where we are looking at microbial assembly by dispersal and examining physical gradient against the chemical gradient of what it's exhibiting. So it's combining the physics and the resources idea, where what cohort of microbe is competing for what resource, and also to metabolically identify the pattern that each of these cohorts exhibit. So it is also something in terms of knowledge gap that we need to look at, where we want to see if mass spec is a universally feasible tool that can be equipped in terms of such studies, because we had Peter explaining the metabolic pattern for these microbes that go along with where they exist, whether that could be used for future studies on a consensus basis. The other things that were looked at were the effects of the quantification pattern. So looking at the viable versus non-viable taxa, and also to have a clear picture of the absolute abundance of the microbes that we are testing or we are examining in these environments. So there needs to be robust methods developed that can bring out the quantification aspect and not only have a qualitative basis. So the other system, model systems component was raised by Janet, where we need to examine if there are model systems akin to what you have in plant systems that can transpose what you see in a field condition where you have a cohort, a specific bacteria that can coexist with fungi that is of symbiotic interest. And there have been reports that in certain buildings you have fungi that can over-dominate and cause health effects. And some of the health effects discussed were wheezing in infants and also asthma, where there has not been a clear correlation effect of what causes those conditions in these populations. And that needs to be addressed in terms of the knowledge gap. The other aspect that was looked at is to look at co-evolving and co-adaptation mechanisms, where we are talking interaction between the three domains, bacteria, fungi, and archaea, and that also needs to be addressed across the platform in a built environment setting. And examining aspects like stress that Venkat brought up with regard to the astronauts, whether there is something that can be correlated in an indoor setting or in a built environment setting of stress-associated microbial increase in, say, patients in specific wards of hospitals. And that is also something of interest. So there is also example that was addressed with regard to examining glucocorticoids that Scott Kelly brought up, where these are stress hormones that are enhanced in conditions that could be as a tracer or a marker that could be examined and looked at. And you could have specific approaches to apply and infer if these metabolites are either increasing or reducing themselves in abundance. And besides that, it was basically to have a synergy of these knowledge gaps as well as the technology gaps that need to be put together so that we have a clear-cut understanding of how we characterize these environments and establish a standard where we can basically integrate data between different diverse environments. So that was what was discussed. Thank you.